Welcome everyone. Today we're diving into Module 2, Chemistry of Nutrients. We'll be focusing on Study Session 1, Chemistry of Sugars I. This is part of BCHM 241, which covers the biochemistry of macromolecules. I'm excited to take you through the fundamental concepts of sugars and their role in nutrition. We'll explore the chemical structure, properties, and significance of sugars in biological systems. So, let's get started and uncover the fascinating chemistry behind these essential nutrients. Let's start by looking at what carbohydrates, or CHOs, really are. They are the most abundant biomolecules on Earth, and you'll find them everywhere, widely distributed in both plants and animals. Now, these compounds contain carbon, hydrogen, and oxygen, following the general formula CnH2n, where N is 3 or greater. The name carbohydrate actually means hydrate of carbon, which makes sense because they are polyhydroxyaldehydes or ketones, meaning they have multiple hydroxyl groups attached to aldehyde or ketone groups. Carbohydrates belong to the same class of biomolecules as proteins, lipids, and nucleic acids, and they play vital roles in energy storage and structural components. At their core, carbohydrates are polymers made up of simple sugars, known as monosaccharides. The term saccharide, derived from the Latin word sacchararum, reflects their sweet taste, which is a characteristic feature of sugars. And among these sugars, glucose stands out as the most important carbohydrate, serving as a primary energy source for many organisms. So, understanding carbohydrates is essential because they are fundamental to life, providing energy and forming the building blocks for more complex molecules. Let's talk about carbohydrates and how they are classified based on the number of simple sugar units they contain. Starting with monosaccharides, which are the simplest type of carbohydrates. These are often called simple sugars because they are the most basic form. Importantly, monosaccharides cannot be broken down further into simpler sugars through hydrolysis, meaning they are already in their simplest form. When we classify monosaccharides, one key aspect is their functional group. They can have either an aldehyde group or a ketone group, which helps us distinguish between different types of sugars. For example, if the carbonyl group is at the end of the molecule, it's an aldehyde, whereas if it's within the structure, it's a ketone. Another way we classify monosaccharides is by counting the number of carbon atoms present. This leads us to terms like trioses, tetroses, pentoses, hexoses, and heptoses. These names tell us how many carbons are in the sugar molecule. This classification is important because it influences the sugar's properties and its role in biological processes. Understanding this classification helps us see how simple sugars are fundamental building blocks in nutrition and biochemistry, connecting to the broader theme of how complex carbohydrates are formed and utilized in the body. Let's take a closer look at some key functional groups in carbohydrates. First, we have the ketone functional group, which plays a crucial role in the structure of certain sugars. Then, there's the aldehyde functional group, another important component in carbohydrate chemistry. Now, moving on to specific examples, diglyceraldehyde is a good example of an aldose, meaning it contains an aldehyde group. On the other hand, dihydroxyacetone is a ketose because it contains a ketone group. Both of these are monosaccharides, which are the simplest forms of carbohydrates. They are essentially polyhydroxy compounds, meaning they have multiple hydroxyl groups, and they have 3 or more carbons. This makes them the basic building blocks of more complex carbohydrates. Because of their small size, monosaccharides are very soluble in water, which is important for their biological functions. To sum up, monosaccharides are polyhydroxy ketones, or aldehydes with at least 3 carbons, and they are fundamental to carbohydrate structure and metabolism. Today, we're going to take a closer look at the structure of aldoses. Aldoses are a type of monosaccharide, and understanding their structure is key to grasping their function and behavior. In essence, aldoses are characterized by having an aldehyde group at the end of their carbon chain. This aldehyde group is what distinguishes them from other sugars, such as ketoses, which contain a ketone group instead. As we examine their structure, you'll notice that aldoses typically have several carbon atoms, with the aldehyde group at one end. The rest of the molecule consists of hydroxyl groups attached to the other carbons, creating a chain that can exist in linear or cyclic forms. It's important to remember that the configuration around each chiral center influences the sugar's properties and reactivity. These configurations can vary, leading to different stereoisomers, which are crucial in biological systems. Understanding the structure of aldoses helps us appreciate how these sugars participate in metabolic pathways and how their shape and functional groups determine their interactions within the body. So, in summary, the structure of aldoses involves an aldehyde group at one end, multiple hydroxyl groups along the chain, and stereochemistry that impacts their biological activity. Let's take a closer look at the structure of ketoses. As you may recall, ketoses are a type of monosaccharide characterized by the presence of a ketone group. This structural feature is what sets them apart from aldoses, which have an aldehyde group instead. The key point here is that in ketoses, the ketone group is typically located at the second carbon atom, hence the term ketose. Now, this structural arrangement influences the molecule's reactivity and its ability to undergo various transformations. Additionally, ketoses often have multiple hydroxyl groups attached, which makes them highly soluble in water. This solubility is important because it affects how they are transported and metabolized in biological systems. Understanding the structure of ketoses is fundamental because it helps us grasp their role in metabolism and how they participate in different biochemical pathways. So, in essence, the defining feature is the ketone group at carbon 2, and this structural configuration impacts their chemical behavior and biological functions. So let's talk about disaccharides, which are essentially the condensation products of two monosaccharide units. That means two simple sugars come together and form a larger, more complex sugar. For example, when galactose and glucose combine, they form lactose, which is the sugar found in milk. Similarly, glucose and fructose join to make sucrose, which is what we commonly refer to as table sugar and is found in sugar cane. And then, when two glucose molecules come together, they form maltose, which is often produced during the breakdown of starch. These disaccharides are important because they are common dietary sugars, and they play a crucial role in our nutrition. Lactose, for instance, is the primary sugar in milk, while sucrose is a universal sweetener. Understanding these combinations helps us see how complex carbohydrates are built from simple units, connecting to the bigger picture of carbohydrate digestion and metabolism. They are condensation products of 3 to 10 simple sugars. For example, you might think of trisaccharides, tetrasaccharides, pentasaccharides, and so on. These molecules are quite versatile and play important roles in our bodies. One key function is as recognition molecules on the membrane surface, often in the form of glycoproteins or glycolipids. This means they help cells recognize each other and communicate effectively. Additionally, they have a structural role, especially glycosaminoglycans, which provide support and resilience in tissues. Overall, oligosaccharides are not just simple sugars. They are crucial for cell recognition, signaling, and structural integrity in biological systems. Polysaccharides are quite fascinating because they contain more than just a few residues, ranging from just over 10 up to millions. This huge variation in size makes them incredibly versatile. Now, when we classify these molecules, we generally divide them into two main groups, homopolysaccharides and heteropolysaccharides. Homopolysaccharides include well-known examples like starch, glycogen, cellulose, chitin, and inulin. These are made up of a single type of sugar monomer. On the other hand, heteropolysaccharides, such as agar and glycosaminoglycans, consist of different types of sugar units, giving them more complex structures. We can also categorize polysaccharides based on their function. Some serve structural roles. Think of cellulose in plant cell walls or chitin in fungal cell walls and insect exoskeletons. These are essential for providing support and rigidity. Others are primarily used for storage. For instance, starch in plants and glycogen in animals act as energy reserves, ready to be broken down when needed. Understanding these differences helps us appreciate how diverse and essential polysaccharides are in biological systems. They are not just simple sugars, but complex, multifunctional molecules that sustain life in many forms. Let's take a closer look at the structure of glucose. Glucose is a simple sugar, but its structure is quite important because it influences how it behaves in the body. At its core, glucose is a 6-carbon molecule, which makes it a hexose sugar. These carbons are arranged in a specific way that allows it to form both open-chain and ring structures. In its open-chain form, glucose has an aldehyde group at one end, which is crucial for its reactivity. This aldehyde group can react with hydroxyl groups on other carbons, leading to the formation of ring structures, which are more common in biological systems. Most of the time, glucose exists as a ring, specifically a 6-membered ring called a pyranose ring. This ring is formed when the aldehyde reacts with the hydroxyl group on the 5th carbon, creating a stable cyclic structure. Understanding this structure helps us grasp how glucose interacts with enzymes and other molecules, and how it can be converted into energy. It also underpins many reactions in carbohydrate metabolism, which is a key theme in this presentation. So, in summary, glucose's structure, a 6-carbon backbone with the potential to form ring structures, is fundamental to its function in biological systems. Let's talk about isomerism, which is when compounds have the same chemical formula but different structural formulas. This is important in glucose because there are 2 main types of isomerism we focus on: D and L isomerism. The key factor that determines whether a sugar is D or L is how the H and O groups are oriented around a specific carbon atom. This carbon is the one next to the terminal primary alcohol group, which is farthest from the functional group. If the O group on this carbon is on the right, the sugar is classified as a D isomer. Conversely, if the O is on the left, it's an L isomer. Most naturally occurring monosaccharides, like glucose, are D sugars. An interesting point is that sugars which are mirror images of each other are called enantiomers. This mirror image relationship is a fundamental aspect of stereochemistry in sugars and helps explain their diverse properties. So, in summary, understanding how the O groups are oriented around this key carbon helps us distinguish between D and L isomers, which is crucial for grasping the structure and function of sugars in biological systems. Today, we're focusing on 2 sugars that are actually very similar, but differ only in the configuration of the hydroxyl group around a specific carbon atom. This subtle difference has a big impact on their properties and functions. For example, D-glucose and D-mannose are considered C2 epimers because they differ at the 2nd carbon. Similarly, D-glucose and D-galactose are C4 epimers, meaning they differ at the 4th carbon. These variations are called epimers of glucose, and understanding these small differences helps us grasp how sugars can have different biological roles despite their structural similarities. Recognizing these epimers is key to understanding carbohydrate chemistry and how these molecules interact in biological systems. Now, let's explore the process of cyclization in monosaccharides, focusing on how hemiacetals and hemictals form. When monosaccharides, which are simple sugars, are in their linear form, they can undergo a fascinating transformation to become cyclic structures. This happens through an intramolecular reaction where the hydroxyl group on one end of the molecule reacts with the carbonyl group, either an aldehyde or a ketone. This reaction results in the formation of a ring structure, which is more stable and commonly found in nature. In this process, depending on whether the carbonyl was an aldehyde or a ketone, we get different types of cyclic compounds, hemiacetals or hemictals. The key point here is that the formation of these rings is crucial because it explains how sugars exist predominantly in cyclic forms in biological systems. This cyclic form also introduces new stereocenters, leading to the possibility of different isomers, which are important for the sugar's function. So, understanding this cyclization process helps us grasp the structural complexity and versatility of sugars, which play essential roles in energy storage and molecular recognition within living organisms. This transformation from linear to cyclic forms is a fundamental concept that ties directly into the overall chemistry and functionality of carbohydrates. This slide doesn't contain enough information. That's why we're moving to the next slide. Most monosaccharides, including glucose, tend to spontaneously form ring structures in solution. This happens because the aldehyde or keto group within the molecule reacts with one of its own hydroxyl groups, creating a stable ring. When the ring contains 5 atoms, we call it a furanose ring, which resembles the structure of furan. If the ring has 6 atoms, it's known as a pyranose ring, similar to purine. In the case of glucose, more than 99% of molecules in solution exist in the pyranose form, which is more stable and prevalent. Recognizing these forms is crucial because they influence how sugars behave biologically and chemically. Understanding the balance between the furanose and pyranose forms helps us grasp their roles in various metabolic processes. These ring structures are fundamental to the overall structure and function of carbohydrates in biological systems. Let's take a closer look at glucose, which is a classic example of an aldose. As you can see, it's a simple sugar essential for energy in many biological systems. Now, when we talk about glucose, one key aspect is its ability to form rings. This ring formation is a fundamental concept because it changes the structure of the molecule from a straight chain to a cyclic form. The ring structure is crucial because it affects how glucose interacts with other molecules and enzymes in our bodies. When glucose forms a ring, the aldehyde group reacts with a hydroxyl group within the same molecule, creating a stable ring structure. This process is especially important in aqueous solutions, like in our cells, where the cyclic form is predominant. Recognizing these ring structures helps us understand how glucose functions in metabolic pathways and how it's recognized by different enzymes. So, in essence, the formation of the ring structure is a key step in the chemistry of glucose and other sugars, connecting to the bigger picture of carbohydrate function and metabolism. Let's take a look at a ketose, specifically fructose. Fructose is a simple sugar, or monosaccharide, that falls into the ketose category because of its chemical structure. The key characteristic here is the presence of a ketone group, which distinguishes it from aldoses that contain an aldehyde group. Fructose is found naturally in fruits, honey, and some vegetables, making it a common source of dietary sugar. It's often used as a sweetener in processed foods because it's very sweet and metabolized differently than glucose. Understanding fructose as a ketose helps us appreciate the diversity of carbohydrates and how their structures influence their functions and metabolism. This connection to the overall theme highlights the importance of molecular structure in determining the behavior and role of sugars in biological systems. Now let's look at how cyclization occurs through intramolecular hemiacetal or hemictyl formation. When the sugar molecule cyclizes, the carbon at position 1, C1, becomes a chiral center, known as the anomeric carbon. This is a crucial point because this new chiral center defines the configuration of the sugar. The anomeric carbon has an O group attached, and this group can be oriented either above or below the plane of the ring, which leads to different stereoisomers called anomers. If the O group on the anomeric carbon is on the right side of the carbon chain in a Fischer projection, we refer to this as the alpha or alpha ionomer. Conversely, if it's on the left, it's the beta or beta ionomer. Understanding these configurations is important because they influence the properties and reactivity of sugars. The term anomers refers to these specific isomers that differ only at the anomeric carbon, and recognizing this helps us understand how sugars can exist in different forms, which is key in many biological processes and chemical reactions. This concept ties back to the overall theme of stereochemistry and how molecular shape impacts function. Now, let's explore why glucose solution is often referred to as dextrose solution in clinical terms. You might wonder, what's the difference between glucose and dextrose? Well, technically, glucose is the name of the sugar itself, which is a simple carbohydrate essential for our body's energy. Dextrose, on the other hand, is just a specific form of glucose, specifically the D-isomer, which is the naturally occurring form found in our bodies. In clinical settings, when we talk about dextrose, we're emphasizing the chemical form that's used in medical preparations. It's often called dextrose because it's the form that's most stable, pure, and suitable for intravenous use. So the key point here is that dextrose and glucose refer to the same sugar, but dextrose is the term used in medicine and pharmacy contexts. This distinction helps ensure clarity and precision when administering solutions, especially in IV therapy. Connecting this to our overall theme, understanding this terminology helps us better grasp how these solutions are prepared, labeled, and used safely in clinical practice. It highlights the importance of precise language in medicine to avoid confusion and ensure optimal patient care.